Thank you for joining us on Coffee Break Webinars. Our topic today is modeling epilepsy at the Institute for Genomic Medicine. 65 million people worldwide suffer from epilepsy. Epilepsy is characterized by seizures, which are the result of excessive nerve activity in the brain. This condition is a range of disorders that varies from mild to severe, some of which can be life-threatening. While treatments are available, finding the optimal medication for any patient involves a period of trial and error, during which both doctor and patient will try to find a solution that improves their quality of life. Unfortunately, for about a third of them, the prescribed treatment will not work. We need to better understand the underlying causes and develop better treatments for those suffering from epilepsy. In today's Coffee Break webinar, Mike Boland and Melodi McSweeney discuss how they are harnessing the power of genetic engineering in mice to explore the neuronal networks behind epilepsy. With a better understanding of these brain networks, it could be possible to classify different types of epilepsy, allowing for the most effective medication to be prescribed for the first time. Without further ado, I'd like to introduce you to today's researchers. Dr. Michael Boland is an assistant professor at Columbia University, where he serves as director of the Cellular Models of Disease Initiative with the Institute for Genomic Medicine. Melodi McSweeney is a PhD candidate currently studying genetics with an emphasis on neurodevelopmental disorders. Both she and Dr. Boland have worked extensively with David Goldstein, as all three of them dig deeper into the medical mysteries of neurodiseases.
Thanks for the introduction, Melissa. As mentioned, Melodi and I are based at the Institute for Genomic Medicine at Columbia University. Columbia University recently introduced precision medicine initiatives in 5 key areas of medicine: undiagnosed childhood diseases, kidney and liver disease, maternal fetal medicine, ALS, and epilepsy. At the heart of each of these initiatives is the Institute for Genomic Medicine, which was created to serve as the face of these efforts. The IGM, under the direction of human geneticist David Goldstein, uses state-of-the-art whole exome and whole genome sequencing coupled with advanced bioinformatics to accurately identify disease-causing genetic variants. The IGM is a highly collaborative multidisciplinary institute comprised of genetic counselors that interface with patients, clinicians, and IGM leadership. It also consists of a genomic sequencing core facility and a team of bioinformaticians. Additionally, the IGM possesses a disease modeling group that focuses on neurodevelopmental disorders such as autism, intellectual disability, and epilepsy. We use human-induced pluripotent stem cells derived from patients as well as human iPS cells and mouse lines generated by genomic editing to accurately represent the genotype of patients. These models are subjected to a number of technical platforms that are designed to identify and characterize genotype-specific phenotypes. Some of these platforms are shown here. Our disease modeling group collaborates with other academic groups and industry leaders to identify and develop new therapeutics. Here, we'll focus on using microelectrode arrays to study activity patterns of primary neuronal networks from mouse models of epilepsy. The IGM, under the direction of Wayne-Frankel, has developed 14 mouse models of epilepsy from genes that affect a range of biological processes, such as the function of neuronal ion channels or transporters, the endocytosis of synaptic vesicles, synaptic function, signaling through cell surface receptors, and RNA processing. We've developed corresponding human iPS cell models for many of them to more accurately study cellular phenotypes. We use microelectrode arrays as the primary screening tool to examine neuronal networks from these models for genotype-specific phenotypes and for recurrent phenotypes across models. Briefly, MEAs are tissue culture wells embedded with multiple electrodes that measure local field potentials generated from electrically active cells. Software then converts that information into an activity profile for each electrode. We use Axion's multiwell MEA Maestro system equipped with a Lumos module for optogenetic manipulation of networks. MEA has a number of advantages over traditional whole-cell patch clamp electrophysiology. It represents a non-invasive method to record network activity from populations of neurons rather than single cells. It allows for a researcher to monitor network development and establishment, and also to test several experimental conditions in parallel. The medium throughput of the multiwell format allows for small-scale compound screening or testing. The IGM has developed a custom MEA analysis package that's freely available to the research community and can be found at the web address at the bottom of the slide.
As a proof of concept, we investigated whether changes in excitability could be identified using the MEA following modulation of neuronal network activity. A number of recent studies have implicated certain microRNAs in epilepsy. For example, when the expression of microRNA 128 is knocked down, it results in seizures in mice and death within about 3 months of age. We used a lentivirus to deliver a microRNA inhibitor called a sponge RNA, which is comprised of tandemly repeated sequences that are partially complementary to the mature microRNA. We used the sponge RNA to knock down expression of microRNA 128 in primary mouse cortical neurons, and then investigated changes in network excitability. A schematic of the microRNA sponge is shown on the left. It is expressed downstream of a GFP reporter gene. On the right, we see cells expressing the green fluorescent protein on an MEA well. The video is an example of recorded network activity. Each box represents one well of a 12-well MEA plate. Each dot is one of the 64 electrodes in each well. The intensity of the colors reflect the activity of the nearby neurons. You can see that the 6 control wells on the left fire much less frequently than the wells treated with the microRNA sponge on the right. A descriptive way to visualize this data is to utilize raster plots, which demonstrate activity from each electrode over time. We generated raster plots using our in-house MEA analysis package to show that in the control wells, there are well-defined bursts that synchronize across most electrodes and well-defined interburst intervals in which there is no activity. The networks treated with the microRNA sponge, shown on the right, fire more frequently and have little and highly variable time between bursts. We further analyzed the activity data using our pipeline and identified significantly increased mean firing rate, burst rate, and synchronous network spikes in wells in which microRNA 128 expression was reduced. We conclude that suppression of microRNA 128 expression results in network hyperexcitability in vitro. Given this proof of concept data, we moved on to test this paradigm by evaluating primary neuronal cultures on MEAs using one of our genetic mouse models. This spider plot shows a clear fingerprint of genotype-specific activity patterns in one of our epilepsy models. When compared to wild-type non-epileptic cells, shown here in black, and given the value of 1, the mutant network, shown here in blue, has a similar number of active electrodes and mean firing rate, which are measures of network health and activity. The mutant networks, however, have significantly increased number of bursts, decreased percent of spikes in bursts, mean duration of burst, and mean interburst intervals. This means the epileptic network has many short, very rapid bursts with very little time between them. The mutant network also has increased number of network spikes and percent of spikes in network events, which are features of synchronicity. This illustrates hyperexcitability and hypersynchronicity in the mutant. Raster plots nicely illustrate this aberrant network activity. On the left is a representative activity plot from a wild-type network compared to a representative mutant network on the right. Both types of networks possess defined bursts and interburst intervals, but the mutant networks exhibit shorter bursts with very short interburst intervals. Also note that the bursts in the mutant network have a fixed burst duration and rigid periodicity in comparison to controls. We recognize this phenotype from our experiments involving pharmacological manipulation of neuronal subtypes in networks. In particular, this phenotype is reminiscent of GABAergic inhibition. when neurotransmission in wild-type networks is modulated by GABA-A receptor antagonists, such as gabazine, we observe significant increases in mean firing rate, bursts per minute, and percent of spikes in bursts, which is reminiscent of our epilepsy mutant networks. These data provide some insights into the underlying mechanism responsible for the aberrant phenotypes observed in the mutant networks. Ultimately, our goal is to use MEAs to identify small molecules or drugs that are capable of correcting or ameliorating mutant phenotypes so they resemble wild-type activity. We've shown that MEAs represent a viable tool to identify and analyze genotype-specific network phenotypes. Furthermore, orthologous disease models based on solid human genetics represent better models for mechanistic studies and therapeutic design. The multiwell MEA platform is amenable to medium throughput drug screening, and lead candidate compounds from large screens can be prioritized based on MEA findings, and these findings can be used for phenotype correction or amelioration before being tested in an animal.
We'd be remiss if we didn't acknowledge David Goldstein, Aaron Heinzen, and Wayne Frankel, as well as other members of the IGM and funding agencies. Thank you for your attention.
And that wraps up today's coffee break webinar. If you have any questions you would like to ask regarding the research presented, or if you are interested in presenting your own research with Microelectrode Array technology, please forward them to coffeebreak@axionbio.com. For questions submitted for Dr. Boland and Melodi McSweeney, they will be in touch with you shortly. Thank you for joining in on today's coffee break webinar, and we look forward to seeing you again.